Today we're diving into the impact of insecticides on plant health, specifically Brassica rapa, known as Wisconsin's fast plants. Insecticides are widely used in agriculture to boost crop yields by controlling pests and weeds. According to Akhtar et al. 2009, while insecticides can provide significant benefits, studies show they can harm non-target species, including plants themselves. Also, according to Pathak et al. 2022, insecticide runoff can damage plant roots, reduce photosynthesis, and limit water and nutrient uptake, leading to stunted growth. Due to these findings for our study, we're using Wisconsin fast plants, a fast-growing species in the mustard family, as a proxy to understand the effects of insecticides on crops like turnips. According to Bird et al. 2017, the crop species Brassica rapa L has significant economic and ecological importance around the world. The domestication and improvement of crops has led to significant phenotypic diversity, resulting in subspecies utilized for oilseed production, human food, and livestock fodder. By exposing Wisconsin fast plants to different concentrations of insecticides in a controlled environment, we will assess how these chemicals influence early growth and physiological health. Our hypothesis is that increased use of insecticides by farmers is affecting plant growth and physiology negatively by inhibiting photosynthesis and preventing an uptake of nutrients needed to survive and grow. This study will help us better understand how insecticides might affect not only crops like turnips, but also broader agricultural sustainability.
To begin, we divided moist soil into 4 specialized planting units, with each unit consisting of 32 cells. We placed a wick in each cell to promote consistent water absorption from the reservoir below. Then, 2 to 3 seeds were added to each cell, which were placed carefully on top of the moist soil. We added 2 drops of water on top of the seeds in each cell to promote germination using a disposable pipette. In order to maintain consistent environmental conditions such as oxygen, light, and temperature, we placed the planting units in a controlled environment in a biology lab room. We kept the planting units under a plant growth light, which remained on at all times. Every day, we observed the plants for signs of growth and ensured they maintained proper moisture levels by watering them each day with 4 drops of water, which is 0.2 milliliters, using a disposable pipette. After 4 days, we thinned the plants, leaving 1 healthy sprout per compartment to ensure adequate space and resources for growth and to prevent competition among plants. Following this step, each treatment group contained 32 plants, resulting in a total of 128 plants across all groups. We then labeled each plant to accurately track individual growth and progress. After 5 days of growth, we began measuring the height of the plant every other day and applying the corresponding insecticide concentrations, which were 0%, which was our control, 1.5%, which was the low concentration, 3%, which was the medium concentration, and 5%, which was our high concentration, to each plant group to simulate multiple runoff incidents.
Significant differences in plant health were observed among Brassica rapa plants treated with varying concentrations of spectricide. In the control group, leaves remained a healthy green with no signs of stress. However, at low concentrations, slight browning appeared at the edges of lower leaves by the second week, indicating mild stress. Medium concentrations caused more pronounced symptoms, including browning and yellowing, which became evident as the study progressed. At the highest concentration, plants exhibited the most severe damage, with widespread browning, curling, and more pronounced veins, indicating a sharp decline in overall plant vitality.
Despite differences in plant health, growth rates remained relatively consistent across all groups. Although stunted growth was not observed at any concentration level, the control group demonstrated the lowest growth over the 8-day period. Meanwhile, the 3% concentration group achieved the most significant growth. Across all groups, plant height steadily increased each day.
We rejected the initial portion of our hypothesis stating that insecticide would affect growth of the plants and accepted the portion that stated the insecticide would negatively impact plant physiology. Both the control group and groups infected with insecticide continuously increased in height throughout the duration of our study. Growth was not stunted, allowing us to infer the insecticide did not affect growth. However, we did notice wilting in plants infected with high and medium concentrations of insecticide, even though they kept increasing in height. According to the growth manual of Wisconsin fast plants, at the 1-week mark, 2 cotyledons will be present, which mirrored our findings and timeline. However, the manual stated that from days 21 to 40, significant damage to the cotyledons and death will occur. In our study, the deterioration of the cotyledons and death occurred at the 2-week mark in plants infected with varying insecticide concentrations, but not in the control group, allowing us to infer insecticide had a negative effect on the plant's physiology. Our research project is ecologically significant because it demonstrated the effects of insecticide concentrations on Brassica rapa, a proxy organism that plays a crucial role in Bridgewater, Virginia ecosystem. The study conducted helps provide insight into how the chemical exposure influenced plant growth and health over a 2-week period. Understanding these effects was essential to assessing how insecticide runoff from local farmers can have a negative effect on local plant health, showing the overuse of insecticides can lead to a negative impact on non-target species, a disruption in food chains, and have a negative effect on potential pollinators.